Everybody, in this video I am going to try and explain this cellular process called nonsense mediated decay, or simply NMD. Okay, so maybe if you watched the previous video, you have an idea of what nonsense-mediated decay is, and now I'm going to try and explain some details about the mechanism. So I want to start with a diagram of, let's say, of a messenger RNA. So let's see. So let's start with the 7-methylguanosine cap over here. And there's the messenger RNA, and here's the poly A tail, about 250 nucleotides long. So let's say the stop codon is down here. The start codon for translation is up here. And let's say this messenger RNA has 2 exon-exon junctions. So these are the places where the exons have been spliced together. So let's say the gene for this messenger RNA has 2 introns, and those introns were removed. So one was right there, and the other one was right there. Spliceosome removed those and spliced those exons together. So this is a gene with 3 exons: exon 1, exon 2, and exon 3. Now the spliceosome will deposit exon junction complexes, and I'm going to name some of the proteins in the complex in a moment, but for now let's just cover this NMD process conceptually. And so here I have an exon junction complex with, let's say, 4 proteins in there, and a large number of proteins, say, in this pink circle here. And the reason I diagrammed it like that is researchers who focus on the exon junction complex consider 4 or 5 of the proteins to be core proteins and the others to be sort of accessory proteins. And there are a lot of proteins, and we're not going to name all of these. So my exon junction complex looks like that. And I'm going to put one over here too, because the spliceosome is going to deposit one of these complexes upstream of every exon-exon junction. So it's deposited after the intron is removed from the nascent RNA. And I use some colors below too, where I have some of these already diagrammed, and they might not match up, but that's okay. Okay, so where are these deposited? So these exon junction complexes, EJCs, they are deposited about 25 nucleotides upstream of the exon-exon junction. So they don't sit right over the exon-exon junction. They're deposited about 25 nucleotides upstream of the exon-exon junction. So now these exon junction complexes are installed when the introns are removed from the nascent RNA, and they will remain on that messenger RNA, which is, when the messenger RNA is a complex with proteins, it is typically referred to as an MNRP for messenger RNA plus proteins, RNP being ribonucleoprotein. So I'll probably just call it an mRNA though. So when the messenger RNA is usually complexed with proteins, in this case the exon junction complex is. So this messenger RNA will be exported out of the cytoplasm, out of the nucleus into the cytoplasm with the exon junction complexes associated with the messenger RNA, and it will be translated by a ribosome. And that first ribosome to translate the messenger RNA is called the Pathfinder ribosome. So the Pathfinder ribosome, what it does is it translates, and as it's translating, so there should be no stop codons inside between the first stop codon and the last stop codon right here. So let's see, this is the first stop codon, this is the last stop codon. There should be no pre-termination codons in that messenger RNA. As a result, the Pathfinder ribosome will travel down that messenger RNA, translating the codons, making the proteins, and it will dislodge the exon junction complexes. So after the first round of translation, there are no more exon junction complexes associated with the messenger RNA. So how does this relate to nonsense mediated decay? So if there is a pre-termination codon, let's say RNA polymerase made a mistake when it was transcribing the DNA, and now there's a pre-termination codon in the messenger RNA. What will happen? So let's say there is a UAG codon located right here. Now that would be a pre-termination codon. So this ribosome would hit that pre-termination codon and something interesting will happen. So let me zoom in up here. So let's just take a closer look at that region. There's my exon junction complex, and let me put a couple of different proteins in here to make it look kind of nice. Okay, and there are a lot of other proteins in here. Here's the exon junction complex. Let's say here is the pre-termination codon. Let's say the codon before is an AAA. It just made one up. So when the ribosome encounters that pre-termination codon, so there are a number of factors that are going to associate with the ribosome and attempt to terminate translation. So there are no transcription factors for the termination codons. Instead, they're recognized by release factors. So let's say this release factor right here is called eukaryotic release factor 1. Okay, that will interact with the UAG stop codon. And so there's another release factor which interacts with that. And then there are going to be 3 other proteins, and I'll name some of these in a moment. And they're different, so I'll use different colors here. Well, I mean 4 different proteins. At least 4. And again, I'll name these in a moment. This is going to be called the SURF complex, which interacts with the eukaryotic release factors. This is, the green one is ERF3, eukaryotic release factor 3. Okay, so when this ribosome terminates, this complex of proteins will assemble with the ribosome, and if it detects an exon junction complex downstream of the termination site, it will trigger messenger RNA decay, which is just another way of saying it will cause the messenger RNA to be degraded. So why does it do that? You know, what is the purpose? So one thought is it's a quality control mechanism. So let's go back up to our model of a messenger RNA. See how this stop codon here is located in the last exon? So stop codons in eukaryotic messenger RNAs are almost always found in the last exon of the gene. So we should almost never see an exon junction complex downstream of a stop codon, right? Because exon junction complexes are installed upstream of introns, exon-exon junctions, and if the translational stop codons are always found in the last exon, exon junction complexes should never be found downstream of stop codons. Now we'll see some rare occasions where they are in the next lecture, but for the most part they should never be found after a stop codon. So if there is a stop codon upstream of an upstream of an exon junction complex, what that means is probably means that that is a mutation, and that if that messenger RNA was not degraded, then it could be translated by many ribosomes, not just the Pathfinder ribosome. So the Pathfinder ribosome clears out the exon junction complexes and looks for pre-termination codons. If it finds one, it degrades it. If it doesn't find one, well, other ribosomes then start to translate that messenger RNA, and that messenger RNA can be used to make many proteins. So if the Pathfinder ribosome detects a pre-termination codon based on the presence of an exon junction complex downstream of that pre-termination codon, it can degrade the messenger RNA before too many truncated proteins are produced. What do I mean by truncated? So shorter than normal proteins, right? Because the pre-termination codon occurs early. And why that might be important is a short protein could cause a lot of harm to the cell. So say I can give you an example here. Some proteins have the functional domain on the N-terminal end, and let's say an inhibitory domain on the C-terminal end. The inhibitory domain normally inhibits the function until, it's phosphorylated or something like that. If you had a protein without the inhibitory domain, then that protein would be on all the time. So that would be one reason why the cell would want to eliminate the possibility of making too many, or reduce the chance of making truncated proteins. Okay, so before I get onto the details, I just want to name some of the proteins that are involved in the process. I don't like asking you guys to think about too many different protein names, because there's very little chance we will remember these after the course unless you become a scientist who works on nonsense-mediated decay. But before we get there, you might remember in the last video I said, or we found that PTCs, pre-termination codons, more than 50 nucleotides away or upstream of the exon-exon junction. So not the exon junction complex, upstream of the exon-exon junction, trigger nonsense-mediated decay. PTCs less than 50 nucleotides upstream of the exon-exon junction do not trigger. Okay, so why? What's going on there? So essentially, I can, let me scroll down here. So what's going on there? So let me just, I have a habit of saying let me instead of let me. Let me make a couple of quick diagrams here. So there's the exon-exon junction. Now, let's say the exon junction complex is installed here. 25 nucleotides upstream. Of the exon junction complex, so of the exon-exon junction. So the exon junction complex is installed 25 nucleotides upstream of the exon-exon junction. And let's say with this one right here, let's say there's a UAG which is only, let's say, a little bit upstream of the exon junction complex. So let's say this is a total of, let's say, 35 nucleotides upstream of the exon-exon junction. And let's say this one here, this UAG, this is a different messenger RNA with a pre-termination codon much further upstream. Let's say this one is 75 nucleotides upstream of the exon-exon junction. Now, with this case down here, the ribosome will encounter, the Pathfinder ribosome will encounter that pre-termination codon. And okay, so we already went over, we saw what would happen. Interactions between the complex that assembles here and the complex on the exon junction complex will trigger decay of this messenger RNA. And that makes sense because the PTC is, more than 50 nucleotides upstream of the exon-exon junction. Up here, however, this UAG is so close to this exon junction complex, and the ribosome is huge. The ribosome's a huge complex. So by the time this UAG gets into the right position of the ribosome, that ribosome has already bumped in to the exon junction complex and dissociated it from the messenger RNA. So, although this ribosome, well, I should say, although translation terminates at this pre-termination codon, there's no more exon junction complexes associated with that messenger RNA, and so nonsense-mediated decay is not triggered. That messenger RNA can keep getting translated. It's going to keep making truncated proteins. So that's where that distance of 50 nucleotides comes from. The pre-termination codons have to be far enough upstream that that last exon junction complex won't be dissociated before the ribosome translates that pre-termination codon. So I should say this is all these things we talk about in the class. These are always models. And with further research, the model might be refined. We might learn something, completely new. But so far in this course, we always talk about the most current model with respect to these various phenomena that researchers discover. So, okay, so what did, as promised, I want to talk about I want to name some of the proteins involved in this process. If you're not interested in the names, then you can pause the video or stop watching the video and take a look at the notes. But let's take a look at some of these, because some of these proteins are rather important. for various aspects of messenger RNA translation and transport. So here I have a messenger RNA. Up top here, messenger RNA that is being translated by pol 2. So this is the DNA template right here. This right here, these are going to be the introns that are going to be removed by the spliceosome. This right here, this is called CBC for cat binding complex. That little blue thing, that's going to be our 7-methylguanosine cap. So in the next diagram here, RNA polymerase II has finished translating. That, I lost my notes here. RNA polymerase II has finished translating, and here we are going to have PABN, so these proteins, poly A binding protein, the nuclear version of it, PABN, is a protein that binds the poly A tail. Again, we have the CBC complex here, and we have the exon junction complexes here. Now, the exon junction complex has, at this stage right here, we have 5 different proteins. We have a protein called MAG-OH, MLN51, Y14, EIF4A3, and UPF1. And UPF1 is probably one of the most important ones. Did I say UPF1? No, sorry. I meant UPF3. UPF3. So, you can probably see why I'm not going to ask you to memorize these names. They are, there's really no point to memorize those. So, and there are a lot of other proteins in here in the pink area there. So, this whole thing right here is going to be exported. This messenger RNA will be exported to the cytoplasm. So all that stuff is happening in the nucleus. It gets exported through the nuclear pore complex, which is a pore in the membrane, in the nuclear membrane. And when it gets to the cytoplasm, another protein associates with the exon junction complexes. That is called going to be UPF2. Now, when the Pathfinder ribosome starts to translate that messenger RNA, we have the protein here being synthesized. The CBC complex, or cat binding complex, is exchanged for a different protein called EIF4E. The PABN, poly A binding nuclear protein, is changed for PABC, which is the cytoplasmic form of the poly A binding protein. And okay, so the Pathfinder ribosome will translate, and okay, made it by this one, okay. Now here, what I'm showing you is it has encountered a pre-termination codon, and there's still an exon junction complex assembled on the messenger RNA. So what happens? Well, we have that ERF1 interacting with ERF3, and the ribosome is when that stop codon is in one of the sites that is being used by the ribosome for translation. So it's recognized by ERF1, ERF3 interacts with ERF1. Then this other protein's blue, this is going to be UPF1. UPF1 interacts with SMG proteins. SMG. These ones are going to be, I think, 4, 5, and 6. Let me think here. I don't want to get these wrong. Let me pull up my notes. SMG 1, 8, and 9. So, as I said earlier, these will interact with proteins of the Exon Junction complex and trigger messenger RNA decay through a process that involves exchanging some of these SMG proteins for some others, namely SMG4. I lost my pen. What's going on here? Oh no. Okay, lost my pen. Doesn't matter. Will be exchanged for SMG 4, 5, and 6. So that's it, essentially, nonsense-mediated decay. So the last part of the video with all the different protein details we don't really need, but if you were interested, I thought I'd go over them for you. Okay, I think in the next video we are going to take a look at maybe those rare occasions where nonsense-mediated decay is used to control expression of a gene in a way that is desired by the cell. So there are some proteins which should only be translated one time, and nonsense-mediated decay can be used to make sure that gene is only translated one time, or that messenger RNA is only translated one time. And I think we're going to take a look at that in the next video.